Okay. We're ready to go. Thanks everyone for coming today, in person and online on the scope. We have Giovanna giving the talk today on CEMAS. Giovanna did her graduate work at Virginia Tech. After that, she went to Cleveland Clinic for a postdoc, and then also NIH for a few years, where she ended up being a scientist as well. Before coming back to Ohio, working at Muskingum University, and then now back over here at CEMAS, getting a bunch of research on mainly on the cryo microscopes, but a lot of different bio. We've gotten her to work on the SCS a little bit.
Yep. Oh, thank you.
I'll try and get that.
All right. So hopefully everyone in Zoom can hear me. Everyone in the room can hear me. Yep. So I'll just go ahead and get started. All right. So for today's talk, I wanted to give just a broad overview of the types of different cellular studies that we've done here at CEMAS and the different microscopes that we use to complete those studies. I'll talk a little bit about sample prep, like what we need from the users and what we do here at CEMAS, and how we use different microscopes to answer different biological questions. So during the talk, please feel free to interrupt me, ask questions. For those of you in person, you can just raise your hand or yell out. And then for those of you on Zoom, we have the Q&A button, the chat button, and then the raise hand function. If you would like a more in-depth discussion about your particular project, you can stick around after this talk or email me and you can set up a one-on-one session. Again, if you're having any technical difficulties with Zoom, you can contact us with the chat function. We've got Dan V here working on all of our technical issues. Okay, so at CEMAS, we have lots and lots of cutting edge instrumentation. This is a world-class facility and the building was custom built specifically to get the highest resolution possible from all of our microscopes. The microscopes that I'll be focusing on today are this Techni TF20, the two microscopes where I'm instrument manager of, the Glacios and the cryo-EMs, and these two microscopes that are shown here are where I spend most of my time. We've also done some cellular work on the Helios 600 and 650 dual beam systems. And as Dan mentioned, I've started a little bit on SEM, looking at cells. I've looked at cells on our older Quanta system, which we no longer have, and then also on our Quattro SEM, which is a very, very fancy SEM. In addition to electron microscopy, CEMAS also has the capabilities to do beam scan and X-ray tomography, as well as X-ray diffraction analysis. As I'm talking about how great CEMAS is and all the different toys we have in this facility, if there's something that catches your eye or you think, hey, that could be useful for my project, and you're interested in learning more, there's several routes to do that. One of them you're already doing, and that's attending a Beyond the Scope CEMAS Discussion Series. You can also schedule a one-on-one consultation with any of the instrument managers for all of these different instruments. And then once you collect your data and you have all this great data, you're not sure what to do with it, you can offer some assistance with processing your data. And we do have limited remote connection to our data analysis computers. Again, you can contact cemas@osu.edu if that's something you're interested in. During this talk, I'll be referencing some previous Beyond the Scope seminars. And if you're not sure where to find them, you could just go directly to our website, click on About, and then go to this events and click that link. And you'll see all of the previous seminars already listed there. So I think there's some chat questions already.
Oh,
it might be up. Never mind.
I got it.
Okay. I'm trying to advance now. There we go. So Dan already covered this information for me. So thank you again. And for those of you that didn't quite catch it, I know it's been a while since I've given one of these talks. So I had my bachelor's degree in both biology and chemistry at Walsh University. For graduate school, I started at the University of Cincinnati and then moved with my advisor to Virginia Tech. So my PhD is from Virginia Tech in chemistry. And during grad school, I did a lot of fluorescence work and a lot of live and fixed fluorescence studies in cells. I did my first postdoctoral researcher training at the Cleveland Clinic, where I learned more molecular biology techniques, PCR, cloning, things like that. And then my second postdoc was at NIH, and here's where I got into electron microscopy. So both room temperature cryo-electron microscopy, as well as room temperature FibCEM. And that's where I started building on my experience in fluorescent microscopy and started working on correlative light and fluorescent microscopy. So that involves labeling something of interest. So shown here, this is a not real Ebola, fake Ebola viruses labeled with fluorescence. And then we visualize that same exact cell in FibCEM to try to get a higher look at what's happening during viral infection. After my postdoc, I got a job at NCI Frederick as a scientist. And with this job, I mainly moved mostly to cryo-EM work. And then I took a break from microscopy and taught chemistry for 3 years at Muskingum University down the road from here. But then CEMAS acquired Aglatios and Krios, and the temptation was just too strong. I couldn't stay away from microscopy that long. So I came back here, and I'm now doing cryo-EM research at CEMAS. So my goal for today is to give you all a broad overview of what we can do at CEMAS. So to start, I'll talk about room temperature methods, including general sample prep, like what we need from the users and the common questions that users ask. And then I'll show some data from the different microscopes we have here, including SEM, focused ion beam scanning electron microscopy, which is FibCEM, transmission electron microscopy, or TEM, and then I'll talk about some of the correlative light and electron microscopy studies that we've done here. Next, I'll talk about cryo methods, and again, go into general sample preparation, mainly what do the users need to contribute versus what the CEMAS contribute. And then again, we'll go into the different cryo instrumentation that we have here, including FibCEM and TEM, and then the technique of cryo-clem, which is very exciting. So for room temperature sample prep, what do we need from the users? So CEMAS, we have multi-million-dollar microscopes in this facility, but we don't have the capability to actually grow cells here. So we don't have the necessary cell culture equipment. So we rely on our users to grow the cells for us, do your experiments, what you need, treat your cells, how you want them to be treated, and then chemically fix the cells and then bring the cells to CEMAS. Once I have the cells at CEMAS, it usually takes about a week for me to get the cells from their dish or whatever vessel they're growing in into the electron microscopes. So for the first day, I'll rinse off the fixative, stain the cells with heavy metals, and I usually stain overnight. The next day, I'll dehydrate the cells. I have to remove all the water from them and then start infiltrating them with resin. And then after that, I'll put the cells in an oven to cure the resin and embed the cells in this hard plastic. And this is just a typical protocol. Each individual project is tailored specifically to your needs. So certain things can be combined and all done in one day if time is a factor. And then Sarah McCullough's Beyond the Scope talk from December in 2021 goes more in depth with this room temperature sample prep, specifically to different types of resins. So again, we're pretty flexible. We'll tailor our protocols to meet your needs. Once I have the cells embedded in this block of plastic, they can be stored indefinitely. And these cells can be imaged with scanning electron microscopy, focused ion beam scanning electron microscopy, or transmission electron microscopy. So SEM is pretty straightforward. I would just cut a chunk off of that plastic resin block, attach it to an SEM stub, and coat with carbon. And I would do this when I want to check to make sure that my cells are present and the morphology looks okay and that my staining worked. So SEM is good to give very quick checks before moving into the more expensive FibSem and TEM instruments. Additionally, if your biological question is happening at the surface of the cells, SEM is really good for that. And in that case, we don't even need to embed your cells in plastic. We can grow the cells directly onto a surface, critically point dry, gold coat them, and then image on the SEM. So shown here is a bacterial biofilm that was grown on a TEM grid. So let's say now I have my cells, they're embedded in a big chunk of plastic. And I'm interested in getting a 3D overview of my cell. In that case, we can use focused ion beam scanning electron microscopy. So the sample prep for that is a few extra steps compared to SEM. And the shape of the resin block that you're attaching to the SEM stub plays a big role in how stable your slice and view data collection is going to be. So for FibSem work, I don't use carbon coat, I use gold coat just to give more conductivity to the plastic. You'll want to avoid big slabs of plastic because the plastic is not conductive. As you're hitting this sample with electrons, it will begin to charge and cause all sorts of different artifacts in your imaging. So you want to avoid these big slabs if possible. You also want to avoid overhangs like this. So as I mentioned, the samples need to be gold coated before you put them in the FibSem. If there's a type of overhang, gold can reach this surface and this surface, but this surface will be just bare plastic. And that's not going to allow electrons a good path to get to this metal SEM stub. You can fit multiple samples per stub. That's fine. But it's a lot easier on the user and whoever's operating the FibSem if your samples are all roughly the same height. So again, with these big, thick samples and really tall samples, once the sample's loaded in the FibSem, we need to tilt it. And if the sample's thick or tall, we increase the risk of bumping into the SEM pull piece or the gas injection system needle. So you want to avoid doing that. And again, you want to avoid long, skinny samples because they're not easy to gold coat. This is my preferred sample shape. So something nice and flat, close to the SEM stub, so it's easy to tilt without bumping into anything in the microscope. And gold can easily reach all of the surfaces. So once it's in the microscope, again, we tilt the sample and then we use the focused ion beam to mill away part of your cell. And then we use the scanning electron microscope to image. And then we slice and image, and go through sections like that until we can visualize what we need to. So as an example, this sample was prepared by an undergraduate named Caleb working in the CREATES program. So I grew the cells and then he embedded them, stained them, and got them in resin. And then we put them on the Helio 650, where we did this slice and view imaging to generate this image stack. And then from that image stack, we're able to generate a 3D volume and segment out what we're interested in. So in this case, we wanted to look at how the mitochondria were connected in cells with a cancerous mutation versus non-cancerous. And using this technique, we were able to easily do that. Then I'm still at room temperature. I still have cells embedded in a block of plastic. If we want really high resolution data, we can move over to our TEM systems. So the downside of TEM is that there's a thickness limitation. So the sample needs to be 200 nanometers or thinner for us to be able to get good signal to noise ratio in our TEM microscopes. And cells are much thicker than 200 nanometers. So I have these cells, and this is just an example of adherent cells growing on a glass bottom dish. We did all of our processing in the glass bottom dishes, and then we cut little pieces out, glued them on a blank block of resin, and then trimmed them down to be able to fit into our ultra microtome. So the smaller we trim, the easier it is to align to the ultra microtome. So again, Leica, the manufacturers of our ultra microtome, recommends a 0.5 by 0.5 millimeter trapezoid. But I typically use 1 millimeter by 1 millimeter because my resin, I'm very confident that it's cured well, it's not gummy at all, and I can get very nice sections, even as large as this. For single images, I usually cut 70 nanometer sections. If I'm interested in 3D studies, then I cut a little bit thicker, just so I have more cellular context to work with. And in that case, I'll cut 200 nanometer sections. And one thing I always warn users who are using our ultramicrotome, you can set the ultramicrotome to cut sections of whatever thickness you want. So I could set it to 70 nanometers, but that doesn't really guarantee that the actual sections are going to be 70 nanometers because it's going to depend on how well your resin is cured, the condition that your knife is in. So I strongly encourage users to go by color rather than the number that the ultramicrotome is showing. And I know that for single images, I want to get this nice silver gold color. For tomography, I want either a purple or a blue color. Once I have my thin sections mounted on a TEM grid, I'll carbon coat them. Again, the plastic is not conductive. I want a nice conductive sample to give my electrons a nice path to ground. So carbon coat helps prevent beam damage. And then on our TechNI F20, we have this software called cryo-EM, which is very, very useful for imaging large areas of the grid very quickly. So we can get a map of the entire 3 millimeter grid if we wanted to. So in this example, I just mapped out one grid square. And let's say I'm interested in getting a closer look here. So I'll put my green crosshair here, say, go there, and the microscope will drive right there. And it's very well aligned. It's pretty robust. And it's been working well over the past few weeks. We can also add multiple markers. Like let's say I wanted to label every one of these little black dots or every one of these vesicles. We can do that and just have the microscope go to each one, zoom in, and take a higher magnification image all automatically. So in this case, I put my crosshair around here and then generated another map. And then I saw this vesicle attached to the nucleus and thought, oh, that looks interesting. Let me drive the stage there. And again, I put my marker down, drive the stage, take a high mag image, and you could see all the different magnification ranges we can go through just on this one microscope. So in my talk, and as I'm talking, I'm saying like, yeah, we can collect many different images at different places. We can also go to each of those areas of interest and collect a tilt series. So the technique of tomography is where you have your sample loaded in the TEM and you tilt your sample, typically between plus and minus 70 degrees is what we can do in the TechNI F20 if we use a special high tilt holder. And so we'll tilt your sample, take an image, tilt, take an image, tilt, take an image, like that to generate a tilt series. And then we'll align those tilt series into an image stack called a tomogram to get a nice 3D slab about 200 nanometers or whatever thickness your section is of that area of interest. So here's an example of a tilt series collected on our Techni F20. And then after data processing, we can generate this slab. So you use this to see what you're interested in, like if you're interested in virus, nanoparticle drug delivery, mitochondrial morphology, what have you. And then you use your 3D reconstruction to see where in relationship to other things that feature is occurring. So earlier I mentioned CLEM, or correlative light and electron microscopy. We can do CLEM with either FibSem or TEM. Just depends on what question you're trying to answer. The protocol that I have the most experience with is this one, where we grow our adherent cells on a gridded glass cover slip that has these alphanumeric markings. We perform the fluorescence microscopy first, and this would be dependent on our user. So CEMAS doesn't have fluorescent microscopes here. So we either work with CMIF, or a lot of users have their own fluorescent microscopes. So they'll image their cells. You can take high magnification images and get the data you need. But then for CLEM, I would ask users to take lower magnification images in order to see the alphanumeric markings. And then that makes it easier for me to map where I need to go for EM work. I also ask users to pick cells from different areas of the grid and try to avoid picking from the very edges. Once the fluorescence work is done, you can then deliver the cells to CEMAS, and I'll get them prepped for EM work. I'll do all the processing just directly in the glass bottom dish, and then use either a Dremel or cutters to cut away the plastic, and use liquid nitrogen to pop off the glass. So on that note, there's a resin that I recommend to make removing the glass much, much easier. And then for FibSem, we can attach, but just make sure to shave the edges down to get a nice even gold coat. Or we can attach quadrants of that gridded cover slip to an SEM stub. And then for TEM, I use a jeweler's saw to very, very carefully cut out individual squares, mount them, and trim them. There is an alternative method to that. If you have a fluorophore that works after dehydration and resin embedding, we can process your cells, section them, and then image those resin embedded sections on a fluorescent microscope. And then take that same TEM grid and image it in one of our TEMs. So this method also works, but the downside is it's not compatible with all resins and all fluorophores. So for example, I can't use my favorite resin with this because this resin has a lot of autofluorescence. So we would have to, again, tailor this to meet your project needs. And this past year, myself, Sarah McCula, who is another staff member here at CEMAS, and Jeff Tonegas, who is a staff member at CMIF, won a staff development grant to develop more correlative workflows here at Ohio State University. And the one that we chose to pursue is photooxidation of GFP. The reason being is a lot of researchers here already have proteins of interest labeled with GFP. So the way this method works is you'll transfect your cells with their plasmids that encode your tagged protein of interest. You'll get your fluorescence images, and then after you collect your nice fluorescence images, we will add media containing diamidobenzidine to your cells, and then add oxygen to your cells, and then really photo bleach the GFP. As the GFP photo bleaches, it releases free oxygen radicals that react with this DAB and form a polymer. And this polymer shows up as a brown precipitate, and it binds to the heavy metal stains that we use in EM imaging. So wherever we see this brown precipitate, it will show up in EM. One caveat to this is you need a very, very high expression of GFP. And then the photo bleaching of GFP doesn't produce a lot of free radicals. Xu et al. Have developed another tag to try to combat that called mini-SOG. It has the same excitation and emission as GFP, but it photo bleaches a lot faster and generates a lot more free radicals. Again, I should acknowledge that this fluorescence imaging and photo bleaching experiments, a lot of work was done by Sadie Potts, who is an undergraduate technician over at CMIF. So now let's get into cryo methods. So currently, with the freezing systems we have, we're limited to smaller samples, so about 20 microns or thinner. The most important advice I can give to users is not to bend the grids. So the cells that we have the most experience with here at CEMAS are adherent cells grown directly on TEM grids. And if the grids get bent, then unfortunately we can't load them into our glacios or cryos. So if this is something you're interested in and you want to try growing cells on grids, I can provide you with some of these 3D printed inserts. They could fit into a glass bottom dish or into a 6-well plate. And all they do is provide a spot for your tweezers to slide under. So you can pick up grids without bending them. So working with cells growing on grids. You can't use the typical copper grids that we're used to using for a lot of single particle and room temperature work. The mammalian cells will not grow on copper grids. So you have to use gold grids. And you have to work very, very slowly. So I know in cell culture, we're used to transferring milliliters of cell culture media at a time using a automatic pipetteman. I recommend not doing that if you've got cells growing on grids, because it's very easy to flip the grid around and lose track of which side your cells are on. And it's very easy to damage the grid. So it's better to use a micropipetter and just go very, very slowly and try to avoid as many bubbles as you can. If you're doing a transfection or treating your cell with something that's potentially toxic, you have to monitor the cell health very carefully. And even just with light microscopy is good enough. If your cells are unhappy and going through apoptosis, they'll detach from the grids much easier and you'll end up losing a lot of cells during the plunge freezing process. And always, always, always keep track of which side of the grid your cells are growing on. After you get your cells to CEMAS, we usually recommend blotting from behind. So you don't want to touch filter paper directly to your cells if possible, and use longer blot times than we're used to using for some of our other work that we do here. After plunge freezing, you have to work very, very quickly, because the longer your cells are out in the air and not safe, stored away in liquid nitrogen storage, the more risk you have of ice contamination. And then during imaging, with room temperature samples that have a lot of heavy metal stains and a nice carbon coat, we can add a lot more electron dose to that, but cryo samples are a lot more sensitive to dose. So if users are screening cells on a cryo system, they have to be more cognizant of what their dose rate is and remember to blank the beam or close the column valves when they're not actively imaging. Plunge freezing, it could be done in one day, but it needs a lot of coordination between the user and CEMAS staff. So as soon as the cells get here, we want to be able to plunge freeze right away. So the typical workflow is users will let me know approximately what time they'll drive their cells over here, and I'll start cooling down all my equipment so that as soon as the cells get here, in less than 5 minutes, they're already plunged and stored in liquid nitrogen. Once the cells are in storage, they can be stored for several months. We have 2 plunging systems here at CEMAS. One of them is the VitraBot from Thermo Fisher. It's a lot more user-friendly compared to our manual system, and you have better control of how hard the blotting paper is touching your grid and has much better humidity control. But the downside, as you can see here, is it blots your grid from both sides. And where the filter paper touches the surface of your grid where your cells are growing on, It can either damage the cells or it can rip the cells right off the grid in that area. We have this manual plunger, which allows us to blot from behind as seen here. So you're not touching the cells directly with filter paper. But you need a lot more user dexterity to use this. Because again, it's pretty easy to bend grids on the system if you're not careful. And then it's more difficult to control the humidity within that chamber. And the reason why we want good humidity control is to minimize evaporation on your sample, because we don't want the cells to dry out during this process. Once your cells are frozen, we can image them in our Helios 600 system. So here we have a very large cell. And in this case, the user was interested in knowing if the plasma membrane was intact or not. So we were able to do a cryo slice and view study and use automated segmentation software to label the outer membrane. In this case, we found out, no, there's multiple holes happening here, unfortunately. But with FibSem, we were able to actually do that. And again, this is a cell in as close to a native environment as you can get. So no chemical fixation and no heavy metal staining. If you want higher resolution, we can take a cryo sample into our TEMs, but the TEMs have that thickness limitation that I mentioned earlier of about 200 nanometers. So for TEM, you're either limited to the very edges of your cells, but if your event of interest is happening in the interior of the cell, we can still look at it. We would just need to thin that area out, and we can do that again in our Helios 600 system. So in this case, cells were grown directly on the grid, and we thinned out an area of interest, transferred that same grid to our Krios TEM, found the area of interest, and then we're able to collect a tomography data set here. For samples that cannot be grown directly on grids, so like a cell pellet or a piece of tissue, we do have a work in progress. We've recently acquired this Klein-Deek cryomanipulator system, which is like a very tiny pair of tweezers that's in the Helios 600, where we can mill out areas from very thick samples and then use these tiny little tweezers to pluck out that lamella and then transfer it to a TEM grid and then get that TEM grid into one of our cryo-TEM systems. So again, this is still a work in progress, but it's from the initial studies, it seems to work pretty well. So cryo-TEM, again, we're limited to thinner areas of the cells. So this is good for looking at just general ultrastructure. If you're looking at nanoparticle drug delivery, virus infections, it's good to visualize that large particle intracellular trafficking. And we can also look at cytoskeleton rearrangements at the very edges of the cells. So these images were all done on our glacios, and they're just to show you the types of resolution we can achieve in cryo-TEM and what we can see. And again, this is with no chemical fixation and no heavy metal staining. If you're interested in more 3D data, again, we can do cryotomography. And for a more in-depth discussion of cryotomography, you can see Bin's Beyond the Scope talk from 2020. It's a similar process to room temperature. So where we tilt, image, tilt, image, tilt, image to generate a tilt series and then align that into a tomogram. But in the case of cryo, we have to pay more attention to dose, and we don't have heavy metal staining. So in order to get good signal, especially at the higher tilt angles, gold fiducials can be very helpful. So here's an example of a tomogram that was collected on our cryo system, and the cells were prepared by Ji Ying, and she's working with this group from University of Pittsburgh. And they were interested in looking at ribosome-associated vesicles. So here we can clearly see ribosomes. We can see mitochondrion over here, and then we could see the rough endoplasmic reticulum. So again, there's no heavy metal staining involved. So we're not masking features, so we can get very high resolution. And our Krios is also equipped with an energy filter, and this makes a big difference in improving the signal to noise ratio, especially at higher tilt angles. In very recent years, the processes of cryofib milling and cryotomography have come a long way. So over the past 10 years, people have achieved tomography data sets that are approaching the same resolution that can be achieved with single particle cryo-EM, which is a very big deal. And the reason being is just there's more automation, better data collection schemes that minimize dose and radiation damage, and just better hardware. And the Krios here is equipped with all the bells and whistles you could ever ask for. So energy filter, CS corrector, phase plates. So we can get very, very nice tilt series with that microscope. So again, cryo-TEM, we have a thickness limitation. If you want to look at something more on the interior of cells, cryo-STEM is also an option. So we can image thicker samples compared with TEM. So that's the plus side. The downside is it's extremely difficult to get nice STEM images at high magnifications in cryo, and it's due to the radiation damage. So in STEM, we're condensing the beam down to a very small point and rastering it across your area of interest. So that's what these rectangles are coming from. But that point contains very high electron dose, and it burns the sample very quickly. So if a user is interested in cryo-STEM, I recommend that they stay at lower magnifications. If you want nice high magnification, high resolution, then it's better to just stick with TEM. And then we have several works in progress. With cryo-CLEM, we're working with another user who has synthesized some fluorescent gold nanoparticles. And these allow us to track proteins of interest in different microscopes. So here, this is his confocal images. And then we're able to transfer the same grid into our Helios 600. And this is all done in cryo conditions. And then from here, we were able to mill away our area of interest and then put it into our TEM. I'm also working on finding more robust ways to specifically label proteins for visualizing cryo-EM. And this is actually the topic of my microscopy and microanalysis conference talk this year. So I'm using this METTEM system developed by Christina Risco. And what that does is tag a protein of interest with metallothionein, and then we add gold salt to our cells, and it forms these dark nanoclusters. So in this case, I transfected cells with a KRAS protein tagged with this metallothionein, and I know just based on my previous work that KRAS tends to accumulate in the filopodia of cells. So this is still treated with gold salts, but not transfected. And then this is my transfected cell. And I could see in the transfected cell, the filopodia have all these dark clusters where my KRAS protein is accumulating. But again, more work on this, hopefully in the next few weeks. So in conclusion, we have a lot of different and exciting ways to look at cells at CEMAS. You can use room temperature, cryoimaging, or both. And if you have any further questions, please don't hesitate to contact me. Again, thank you all for attending. If you have any other general questions, again, about becoming a CEMAS user, or you're interested in all the cool developments happening at CEMAS, we have a lot of different ways here to connect with us. With that, I'll end here and open the floor to any questions.
So if there's any questions here or online, go ahead, type those in. We can chat a little bit more about it as well.
Yes.
In the skin imaging, is it common contamination or actually damage?
that's a good question.
Cooling down will also make the
Yeah, I think it's a little bit of both, but if I keep the beam on long enough, I could definitely see like bubbling and holes forming. So I think short term it's carbon, but the longer I keep the beam on, the more my sample is melting.
There's probably no way of cleaning it.
Once the sample is plunge frozen, I have to keep it at liquid nitrogen temperatures. But yeah, again, it's hard to say. I do think I can double check, but I'm pretty sure a lot of it is radiation. It's more so radiation. The reason I'm saying that is because I can physically see holes. Like my sample was there and now it's no longer there anymore. Yes.
Okay, so my question is pretty basic, but I'm a relatively new researcher now studying both biology. It's not something I've done before, so I'm trying to figure out ways that I can image my cells and image healing over time. And actually one of my suggestions from my I just got my K award, but the part of the feedback was, oh, you have CEMAS, you should take advantage of it. They have so many different things that would be useful for this study. How would I go about figuring out like ways in which it would be useful for my study and how to design things that I could incorporate all the technology
That's a very good question. And I think the easiest way would be to trying to share my screen here. To just go on our website and there's a link that says become a new user, and it'll ask, it's like a Qualtrics survey form, and you fill that out and tell us a little bit about your project and the questions you're trying to ask. And based on that, our admin team will put you in touch with the instrument managers that they think will be the most relevant. Or you could just email us directly too, and we'll point you in the right direction. Like you can email anyone here or email cmas@osu.edu as well.
I know.
Yeah.
That stuff. We have bio people at Giovanna, Sarah.
Okay.
Very useful.
Yeah,
We like talking about it.
Yeah, and there's a lot of possibilities here. Yeah. Yep.
I should probably note that the micro-electron diffraction, can you tell me a little bit more about that? Is that the rotational, when you take the diffraction pattern and you can tilt the sample, you get the reciprocal space?
Yes, so that capability is on the glacios, and we have the software and tilting up, I should say. I know the software is called micro-ED, and it just tilts and takes a diffraction, then tilts, takes a diffraction, tilts, takes a diffraction. Yeah.
I mean, that's quite big in the pharmaceutical, to solve structures.
Yes, yeah, to get high resolution structures. Yeah. So that capability does exist here.
And you can put any solvent in material solvent. What's the limitations of that?
It has to form a crystal and it has to be a small crystal. So the size of the crystal plays a role. So Nicole Hofer was one of Dave's postdocs who was really instrumental in getting the micro-ED set up and running here. And she was also using cryofib because some of her crystals were just too large to get good signal to noise in the glacios. So I would say that is one limitation. Other limitation in general is just getting things to crystallize and getting things to crystallize to the right size and then getting them on the TEM grid and the sample buffer thin enough to where we can actually get a beam through it. So there are a lot of caveats to that technique, but it's possible. I think Nicole has a few publications submitted where she's shown it's possible and we can do it with the equipment that we have here. Are there any other questions?
Back to the lab. Like what, so you don't have any direct lab stuff, but what would you like to be doing more with the lab? What would be like the most useful thing? So I know you're collaborating a lot with other places, but like,
Yeah,
where could that go? Like the easiest, the instrumentation wise, what would be food wise?
like me personally, if I had an unlimited budget, I would get a cryo-EM fluorescent microscope and just be able to visualize frozen samples in fluorescence and then transfer directly to our cryofib or cryo-EM. Because I think part of what happens is right now we're doing all of our fluorescence work at either room temperature or 37 degrees. And then we have to transport the cells to CEMAS to freeze them. And during that transport, things can change. Like cells can start to ball up if they get too cold, or they'll just start to die or become unhappy. So it would be great to just be able to freeze first, then do the fluorescence imaging, and then do all of our EM work. But that's, yeah, another thing, and actually Jose Smokowski and Dan Huber are working on this, working on a better way to transport cells. So they're building a little portable CO2 incubator so we can keep the cells happier for longer as we're driving them across campus. So I think that will help us out a lot in the short term. Yeah.
How about live animal? So you had a beautiful mouse, for example, and you did something to track it over time without sacrificing the mouse. Is there that capability here?
We don't have live animal imaging here at CEMAS, but there is a live imaging lab on campus. And I can try to find their website and send it to you if you'd like.
I would.
Okay, perfect.
Go back to, so Johan's question, we looked this up, I'll ask you so. So comparing dose from STEM versus TM, how do people mainly compare those? Like, because it's gonna be a focused probe that's for a certain time versus hopefully a random distribution over an area.
Yeah, so Austin and I have talked about this quite a bit. And it just seems there's no like hard and fast rule over what dose is the ideal dose or what dose rate is the ideal dose rate that we want. So a lot of it has just been trial and error as far as like dwell times and things like that go. And then I think Austin actually has a calculation that he uses to calculate the dose per pixel of his cryo-SEM imaging.
Do you do it by pixel or do you just say it's time over, you don't know how large the probe is exactly? I don't think that person has to get before.
Yeah, I can double check with Austin, but I remember trying to calculate it per pixel and then trying to convert that into dose over angstrom squared. Yeah.
I was sure that'd be that accurate because you're averaging over a pixel.
Yeah, you're
actually distributed by the whole pixel.
Yeah, that's common. Yeah, it's going to depend on your beam size, and then again, how fast you're going. There's a lot of factors to consider there. But yeah, maybe.
But like as far as beam damage goes, you have looked at like just relatively like this is how much time, much dose, I get the same amount of beam damage qualitatively between the two.
I haven't done too in depth with that. I just saw once I started increasing the magnification and I saw my sample starting to bubble, I just immediately backed down in magnification. Yeah. Yeah, he helped me a lot as far as like getting the glacier set up for cryo-STEM and helping me like align everything nicely, trying to image the cells without melting them completely.
That microscope.
Oh, yeah.
Eventually.
Yeah, we can try those in the glaciers. It might look
We have all.
Yeah, but we don't have cryo data.
There's cryo data.
Oh, there is cryo. Okay. Yeah, yeah, that's right, because they did cryo-EM work on this stuff. Any additional questions?
See questions online. Any questions, or feel free to hang out, chat with us more afterwards.
Yep. All right. Thanks
Thanks
again for
again.
coming. Thank you.